Okay, welcome everyone. I'm Amrit Mudda. I'm a Professor of Neurosciences, and it's a pleasure to welcome you to this session, this webinar that we've organised for you. I'm just going to give our audience a couple more minutes to join us, but I thought I'd just say hello so you know that we're all here. I'm joined by Professor Max Crispin, who's a Professor of Glycobiology, who will take the session today. I'll introduce him in a minute, and I also have Sarah Dinham, who can answer questions related to perhaps non-academic elements of questions that you may have. But we'll just give it a little bit of time, maybe one more minute. It's 5 o'clock on my clock, but we'll give it one more minute. The numbers are slowly going up. I can see that we've got 53 attendees already. Okay, right. It's 5:01, so I'll start the session. The attendee number is going up too, so we will start. This session is going to be recorded, so for anyone who doesn't get to attend it, if people have registered or have had to leave for any reason, you will be able to see a recording of it afterwards. So let me start again then, for those of you who have just joined us. I'm Amrit Mudda. I'm a Professor of Neurosciences at the University of Southampton in the School of Biological Sciences, and I'm very pleased today to be hosting this session with Professor Max Crispin. This session is designed really to give you a flavour of the type of research that we do in biological sciences. Why is that important for you as potential students, as future scientists? Well, being a biomedical scientist in this day and age is it's always been a very important career choice, but the value to our society has really come forward in the last few months with the pandemic. And you will see from the talk that you will hear from Professor Crispin how what a scientist does in his or her lab can really make a difference to challenges that our society faces. So those of you who are unsure about what career to go down and what to do in the future, perhaps you may be inspired by what you hear today. As well as that, we wanted you to get a feel for the sorts of individuals who are likely to teach you if you decided to do one of the degree programs in biological sciences. We are very keen on research-led teaching. That's very much part of our ethos. So I, for example, am a Professor of Neurosciences and I work on Alzheimer's disease. So I organise one of the modules in the final year, which looks at diseases of the brain, and then I will be giving, and I in fact this morning gave a lecture on a disease that I work on, and my lecture will invariably be related to things that I know firsthand, meetings I've been to, papers I've read, research I'm contributing to. And in the same way, Professor Crispin and the research that he does, any teaching that he does, will be very much influenced by his research. So it's very important for you as the scientists of tomorrow to be taught by the scientists of today. And what better way for us to convey that by giving you an example of a lecture that's given by a member of staff. Okay, so Professor Crispin is a Professor of Glycobiology. He did his first degree in biochemistry, and that was at the University of Oxford, and he continued in Oxford where he did his PhD in biochemistry, but it was a joint position with the Scripps Institute in California, and he then remained in Oxford as a Professor of Glycobiology and joined us about 4 years ago in Southampton. He's done some very impactful work in glycobiology, looking at viral proteins, and that's what he's going to talk to you now for about 30 minutes or so. So I hand over now to Max Crispin.
Wonderful. Well, thanks so much for that introduction. I find this this stage so exciting. I remember vividly when I was choosing what to study, just the sort of excitement. And I have to say, after so many, a few decades later, it hasn't disappointed at all. So at school, you know, you begin to get exposed to different elements of science, but it will capture your interests. You know, the undergraduate course is broad. You'll be, you know, trained in different elements. And what I find, well, what you should find reassuring is that you will quickly realise what talents you have and how it aligns with either future research topics, but for that matter, the skills you gain are very broadly applicable. So I find myself, I read straight biochemistry, but it taught me not only how to deal with mountains of quite complex information and simplify it. I mean, what career doesn't need to be able to do that? But it's also taught me, I suppose, to do things like to write. Now, this isn't so immediately obvious. I thought as a sort of schoolboy, you know, I thought I was going to be good at chemistry. I'd go and do biochemistry, and I wouldn't need to be troubled with things like writing. Whereas actually, you find yourself suddenly being exposed to the full range of skills, and all the degrees that you'll be looking at do equip you for that. So I would say, as my recommendation, is to relax about the future and focus on what you enjoy. And I hope so that I can convey some of that enthusiasm about some of the work we've been doing. Now, as Amrit mentioned, I moved from Oxford to Southampton 3 or 4 years ago now. Just budge that slide ahead. And I mean, Oxford's famously pretty city. What on earth motivated me to move to Southampton? Well, Southampton University is just incredible. The facilities we have are world class. We've got funding attracted from numerous different international levels. I was able to relocate the lab next to a world expert in mass spectrometry, in so-called proteomics. And so I found the environment that I really needed to thrive. And I have to say, it's not just research, it's a lovely place. And so I've really enjoyed the whole offering. And you'll see in the next slide, this is what you enjoy looking out from a nice life science building. Now, I brought a team of scientists with me. This is a picture of Al, he's doing his PhD. And one thing that sort of really motivates me is being able to work with young scientists and share some of the enthusiasm I have with the next generation. I've managed to publish numerous papers with people who were undergraduates at the time. So, you know, the life sciences move quick. And so when we have project students involved, you know, more often than you might be thinking, it actually produces work that is of real importance and value that we're able to publish. And so, you know, it should be really quite motivational hearing those types of stories. It's very fruitful. And one area which we've been focused on that we found particularly productive is the study of types of molecules called glycoproteins that are present on the surface of viruses. So you'll all be familiar with this idea, the so-called sort of central dogma of molecular biology, that genes can make proteins, or here peptides, they can fold to form functional proteins. And, you know, this really has defined, you know, modern molecular biology for decades. But this is an oversimplification in numerous ways. And one aspect I want to highlight to you now is the fact that it isn't just proteins. Proteins are themselves complicated beasts, and they are heavily modified. In this case, they can be modified with carbohydrates. Now, I'm going to use words like glycan and glycosylation, carbohydrate. There's all the same thing, just to illustrate the modification of proteins with sugars. So these are physically covalently bound onto your proteins. And by the end of this session, I hope to have convinced you that they're quite an important factor in considering how viruses evade the immune system, and also an important consideration when manufacturing vaccines. Now, all glycoproteins are manufactured in the sort of secretory pathway of the cell. So here you can see coloured in yellow. This is a very simplified diagram of the cell, but in yellow is the sort of luminal spaces, the secretory system, where proteins can get pushed out of the cell, either on the surface or secreted. So as proteins get pushed into the endoplasmic reticulum and transit into the Golgi apparatus, that's where they're getting carbohydrate added, and famously, as they get really chemically complex. Some of that complexity, this will look slightly terrifying, but carbohydrates are put on in the ER and then undergo a series of enzymatic reactions and get increasingly complicated. Now, if you were to try to understand every single reaction before you proceeded, nobody would get anywhere. So you have me telling you now, you can relax on some of the details. The main transition that's occurring with these glycoproteins is they start out with mannose residues attached, which I've coloured green. There's lots of chopping and changing, and they become increasingly complicated, and we call them complex glycans, and I'm going to colour those in magenta. So it's that transition from simple mannose structures to magenta. Now, with the case of HIV, which of course is the pandemic in HIV is continuing to be a huge disease burden globally. You'll see on the diagram on the left a sort of schematic, if you like, of a typical HIV virus, and it only has a handful of viral spikes that project out of this lipid membrane. Now, when you think of how the immune system recognizes HIV, it's worth keeping that image in mind, because the lipid membrane, this virus is budding from your cells, taking with it your human membrane, so that's immunologically self, and yet it's only got a few of these viral spikes. Now, within the viral spikes, we'll see this movie of the structure of the viral spike, showing the protein in grey in the middle, with the glycans surrounding the outside, shown in green. Now, this is a really important point, because what your immune system sees is really the foreign virally encoded protein. The sugars are added by your own cells, and so are immunologically self. So this is why I like this image of this wolf in sheep's clothing. The actual viral protein wolf is quite hidden amongst this dense array of glycans. Now, one important feature here to realize is that the attachment points of these sugars is driven by the sequence of the protein. So there's a particular amino acid sequence, happens to be asparagine, GAP, then serine or threonine. If that happens in a secreted protein, and a sugar can get added on to the asparagine residue. So the density of the glycans is actually driven by the genome of the virus. So that should prompt some thoughts. Well, what are the selective pressures that's given rise to this? Now, you'll have to take this on trust from me that this is very heavily glycosylated. So there's multiple sugar attachment points compared to what we typically see in human proteins. And what we see is that this is an immune evasion strategy. So by putting on these self sugars onto the protein, it's able to hide its viral protein away from immune recognition. So it's a very difficult challenge, but it's not just the sugars the problem. HIV is famously very diverse in terms of its sequence. So we call it antigenic diversity. And this just goes to show a little diagram illustrating the variation that you can expect to see within a viral population. And here we've got a diagram of HIV diversity from the Democratic Republic of Congo. And on the same scale, we have influenza in Canada, which looks like a dot on my screen. it is virtually non-variant. It'll be just 1 or 2 mutations. And actually, even if you look within one patient, we see that the diversity of HIV actually exceeds that of flu globally. So when we think of how to make a vaccine, well, you're not targeting, in the case of HIV, one virus. It is a swarm of different viruses, and this is what's meant it's been so challenging to target. But this is also, on the other hand, why there's been so much success in targeting the latest coronavirus outbreak, because whilst there are mutations appearing, it's nothing like what we see in HIV. So all the methodology that's been developed to try and develop an HIV vaccine was ported over, and it solved the coronavirus vaccine problem very rapidly. Now, all is not lost with HIV. I should emphasize that there is no HIV vaccine, but there are a number of different sort of hints of solutions, and I'd say stronger than hints. There's certain things which I thought when I entered the field were insurmountable problems. Is the immune system ever capable of properly developing an immune response that could neutralize the virus? Well, the answer is yes. People infected with HIV, about a third of people develop antibodies that can cope with some of that diversity and are broadly neutralizing antibodies. Now, you might think, well, that's great, we've solved the problem. Well, no, for that patient, the diversity is still too large. But those antibodies, if you weren't already infected with HIV, would be capable of protecting you. And amazingly, these antibodies are recognizing the dense carbohydrate shield. Now, the density of the sugars is so high, even though the sugars are human, they're self, the density is sufficiently high that it is being recognized as non-self by the immune system eventually. Now, this is quite a complicated little picture, but I wanted to throw in some sort of technical things, because you can see in blue is the tip of an antibody recognizing in green the viral glycoprotein. And the little rings you can see that it's touching are the sugars, the chain of sugars. And what you'll see is it only makes a very small list of contacts between the blue ribbon antibody and the green ribbon protein. The rest of it's through carbohydrate. And how it's so resilient to mutations is because it's not really relying on binding much of the protein surface at all. And so it takes only particular rare mutations to knock off this recognition. Now, as soon as these types of antibodies were identified, basically the whole field went on a hunt, saying how common is this? I mean, nobody had really thought that you could have antibodies recognizing the carbohydrates, but sure enough, there are now tens and tens and tens of examples of highly potent antibodies, and these are listed in these crazy acronyms. But I've coloured that diagram of the viral envelope glycoprotein in different colours to illustrate where the footprints of these different antibodies. And the take-home message is that the vast majority of the sugars presented on this viral glycoprotein can, in fact, be targeted. So what's the problem? Well, the problem is these antibodies, they can suppress infection if you were to give them as a therapy, but then they can't eliminate the infection. They're only occurring in a limited number of people and only after years of infection. And well, frankly, they're heavily mutated. So antibodies themselves mutate to gain their specificity. And these are quite unusual antibodies. But as I said, this was a challenge I thought may not, this could have been a insurmountable hurdle, but it's not. The immune system can find solutions to neutralize HIV in a useful way in multiple different ways. So the HIV field is now trying to find ways to elicit these antibodies by vaccination, by immunization. Now, to what degree is this shared with other envelope viruses like coronavirus there you see on the far right? I mean, this little panel I've assembled is Lassa fever and Ebola on the left. Those are sort of high containment hemorrhagic diseases. They're viral glycoproteins, all resemble things like influenza, HIV, coronavirus. All of them have this similar architecture. All of them display these similar types of carbohydrates. And so, you know, would some of the tricks used for one, developing a vaccine for one, apply to the others? And what are the lessons? How are they similar? How are they different? You know, to what degree could we ever get, say, a universal therapy or universal vaccine? Now, within Southampton, we've managed to generate a really quite nice array of analytical instrumentation to study glycosylation. So while some of this can be scary, you know, these are complicated, really expensive pieces of equipment. They give out quite simple answers, actually. So these are just an illustration of 3 machines, and the one on the left, there's mass spectrometer, and we use that one to study the sugars attached to particular peptides containing particular sugars. The machine in the center is a liquid chromatography machine, and we use that for studying the overall pool of carbohydrates, which we fluorescently label. And in that little diagram, you can see the green oligomannose glycans, and we're able to measure the abundance of those relative to the other complex structures in magenta. And on the right, we actually have a so-called ion mobility mass spectrometer, which is incredible because it has a gas chamber where big floppy molecules go through slower than molecules of the same mass that are small balls. So in a way, we can separate out molecules based on their shape and get beautiful spectra of carbohydrates, which are often this way of basically separating the signal from carbohydrates from other things. So using this suite of different approaches, hopefully we're back, we can study different HIV immunogens. So these are proteins which are being explored to elicit useful immune, you know, they're candidate vaccines, useful immune responses. So the top is one of the fragments called GP120, and it was known for decades that when you make GP120, you get a population. Now this is a UPLC, this liquid chromatography diagram, showing the different sugars, show you these little spikes, with the green oligomannose population. Now remember, those are the structures that are put on when proteins are first made in the secretory system, and normally they're chopped away. In HIV, they're not. And we can compare that to an artificially trimerized, so you have 3 copies of the GP120, or a native-like trimer. And you'll see on the far right little diagrams, and these are electron micrographs showing the structure, and it's only the bottom one which shows these nice little propeller structures and a sort of native-like fold. And those are the ones that have the most green oligomannose. So it seems to be that when you have correctly folded material, you get changes in the associated carbohydrates. Now, we wanted to study how that these sugars are processed on a site-specific basis. And so on the left, what you have is a comparison of 2 different mass spectrometry techniques measuring the types of sugars attached at particular sites. And this gives you just 3 examples. One of a sample just with complex glycans in magenta, one showing entirely oligomannose glycans in green, and one that's mixed. So this gave us comparing these methods gave us a lot of confidence that that this was quite quantitative method, because these were very different methodologies and they gave the same answer, basically very similar. So we could then use the most convenient one, which is a tandem mass spec, where we put the liquid chromatography machine directly in line with the mass spec, and we can therefore measure the different sugars attached at all the different sites. And we can then plot the results of that onto our structure of the molecule, and thereby seeing this sort of map of different sugars that are wrapping round the viral spike. And you'll all see that there are regions that just green oligomannose structures. So the virus has got sufficiently high density of sugars that the enzymes are not properly trimming back the sugars. And so you get these big patches of mannose, and those are ones that are often targeted by these antibodies, these broadly neutralizing antibodies. Now, when we compare those fragments of the viral spike with the native-like this native-like spike, we can plot where the differences are occurring in terms of their sugar processing. And the take-home message from this diagram is that when you only express one sort of lobe of the trimer, the glycan processing is not resembling the native form at all. And so you get this sort of the overall architecture and packing of the intact molecule is really important in recreating the viral processing. So in here, red and yellow show the increase in the amount of mannose present on the native spike versus the intact, versus the sort of different fragments or artificially here pseudo-trimerized trimer. Now, to really illustrate what, how's that happening, you can actually go to the enzymology. And on the far left is a crystal structure of an enzyme that trims mannose. So this is mannosides, and it's in complex with its target, its green oligomannose target. And you'll see just how extensive this enzyme is, looping and embracing the whole of the target structure. Now, in contrast, this middle panel with the yellow backgrounds are lectins. These are your immune system that can recognize carbohydrates, and they just touch on the very tips. And similarly with antibodies here, you can see the bottom with the blue backgrounds, the antibodies reach in and form quite extensive contacts with carbohydrates. So the take-home message here is that different molecules that need to interact with oligomannose residues do it in completely different ways. And so you can have enzymes failing to process viral glycoproteins, and yet those sugars are still accessible for antibody recognition or indeed cellular receptor recognition. Now, when the coronavirus outbreak happened, so this is a year ago, I was working with my PhD students, and we realized that we could contribute to our understanding of the coronavirus. So we contacted a group in the University of Texas, led by Jason McClellan, and he sent us the gene for the latest, the Wuhan strain of the coronavirus, and also some protein they'd manufactured in the lab. And basically, we were asking ourselves, does the coronavirus have these same mannose signatures compared to HIV? Is it really densely glycosylated, or is it less densely glycosylated and therefore much more, readily recognizable by your immune system? Is there a lower density of the shielding? So we applied the same old technology that we had, and I have to say we did this project in 2 weeks of receiving the material. It was quite full on, but we had all the methodologies set up for studying HIV. So as soon as the outbreak occurred, we were able to just apply the methodologies. So we did a characterization by mass spectrometry of the different sugars attached at different points on the virus, and we're able to produce this big map of the glycoprotein spike, illustrating really only a few spots where there was a lot of mannose. So already you'll be able to look and say, well, actually that map looks very different from HIV. And so we actually had, because those people were so desperate for positive news, that this was actually got in the news. This is in the Daily Mail and in Forbes and various other news outlets, Sky News, and I was even interviewed on Russian TV, the equivalent of Newsnights. And the message here was one of real positivity, that the glycans were not exhibiting such a dense glycan shield, which in the case of Lassa fever, really limits its ability to elicit an immune response. So yeah, we thought that this would be readily neutralized by the immune system, which in practice it was. Now, although that map makes it look like there's not much mannose, when you look at the percentages, actually there is still a degree of lower mixed sites, low amounts of partially mannosilated sites. But we can then take that same methodology and apply it back to that same panel I showed you earlier, and we are able to go, well, where does the coronavirus is here? The SARS, the first SARS outbreak shown on the left, the latest Wuhan strain, the SARS-CoV-2 shown in the middle, and the Middle Eastern respiratory syndrome virus, the MERS, shown alongside the Lassa fever and HIV. And the take-home message is that these coronaviruses, regardless of the different strain you're looking at are show much lower density of glycosylation and show much less shielding compared to HIV. Now, whilst that's moderately abstract sort of concepts, that methodology has also enabled us to look at things like the latest vaccine strains, or vaccine approaches. And we've been working with the team in Oxford University studying the Oxford AstraZeneca vaccine, which I see why people call it the Oxford AstraZeneca vaccine, because its official name is the ChAdOx-N-COVID-19 AZD, etc. vaccine. But this was the official strain. And the question we wanted to ask was, does this sort of adenoviral vectored vaccine give rise to native-like SARS viral spikes? So just to recap, this diagram shows you that how the sort of Oxford adenoviral vaccine works is it takes an adenovirus that's safe, it's put in the gene for just the spike protein of the coronavirus, so nothing else. And so it's safe, it's not going to replicate and give you SARS-CoV-2, but it basically imparts that gene into your target cells, and your own cells manufacture the viral spikes. And so they become little factories, if you like. And we were able to basically show, using all the methodologies I've just described, that the carbohydrate processing resembled that of native virus and these recombinant proteins. So despite the fact your cells are getting infected with adenovirus and all the rest of it, they're able to reproduce native-like spikes. Now, this was also really important because many vaccines have used a little trick to stabilize the trimer. And you'll hear this called like the 2P. They've added 2 prolines artificially into the gene in order to trap it from basically undergoing that fusion event. So it's stabilized. The Oxford vaccine does not have this. And so there's been some speculation that the Oxford vaccine could be somehow improved if it had these stabilizing mutations, and that it might not be generating very good material. Whereas our study was able to reassure people that despite not having these little engineering tricks, it was indeed inducing lots of native-like spikes. And this was of sufficient interest that last week it got into the national press. And I think my little phrase I used of illustrating how your cells turn into little factories was helped capture people's imagination. But really it was the illustration we did of the data. So this is an artist impression of the surface of Oxford AstraZeneca vaccine induced production of little viral spikes, and we were able to image using electron microscopy all these little viral spikes on the surface of cells, which is precisely what triggers your immune system. So I hope in this little talk that I've been able to illustrate how learning technical aspects of biochemistry and how molecules work and behave can really help inform various vaccine strategies sometimes against viruses where there's no vaccine, like with HIV, where it's continued program work, and also in the rapid production of vaccines, and just benchmarking how they're working, and understanding whether further modifications would be productive. So I'll end here with a little picture, thanking people in the lab who've done a lot of the work I've highlighted here in green. These are 2 of my PhD students who did quite a lot of the work I've shown. Yasunori, who was actually got a prize from one of the main publishing houses for being the most cited young scientist this year in biomedical sciences, and he's here working in Southampton, just accepted a job in AstraZeneca as it happens, and shown appropriately socially distanced on the TV screen is Joel, who's just finished his PhD in our department. So finally, I need to also highlight that this work is expensive to do, and that we're really dependent on a lot of international funding bodies. And this particular work was supported by the Bill and Melinda Gates Foundation, the US government through the National Institute of Health, and a variety of HIV programs, including the International AIDS Vaccine Initiative. So on that note, I want to encourage people to be using the Q&A, and Amrit and I will hopefully answer any questions that come your way.
Max, you have lots of questions to answer. I've been trying to answer as many as I could in the chat, but there's a bottleneck of questions to pass over your way. But I guess I'm going to kick off with the first one there that I was just typing an answer to, which I guess it talks to your thank you slide. The question there is from Zainab. I'm sorry if I'm pronouncing it incorrectly. The question is, can students take part in research with professors like yourself while studying in Southampton? And I saw you had Maddie Newby's name there. Yeah. So perhaps you could give a little bit of an example. The answer is absolutely yes, but let's give you real examples.
And let's evidence it with, I mentioned that I published with undergraduates. So this is people who were not just joining labs for a PhD, but actually I've published papers with undergraduates. I did that in Oxford several times, and in Southampton, we've just managed to do that. So only after a couple of years, we've had work of sufficient importance and quality to be published as an undergraduate researcher. So yes, Maddie Newby was a final year student and was working with us during the vacations to help manufacture viral glycoproteins. And of course, when the coronavirus outbreak occurred, we basically encouraged her to finish up her studies as quickly as possible, and we managed to get her working in the lab quite, and became quite an essential part of the team. So she's now, I think it's co-authored 3 papers, and she only finished her studies recently. So yeah, it's tremendous, and there's plenty more on the way. So we've managed to persuade her to stay on for graduate studies. And we also see that's quite a common path, that it's a bit self-sorting really. People volunteer to the lab, get part-time jobs, gain interest, and it's very often that I recruit PhD students from the undergraduate population. So absolutely. Now there's also, that's quite an extreme end. There's a whole other sort of different paths people take, including 4th year projects. So my lab hosts project students, and they work in amongst the team, where it's not just me. There's a group of experienced scientists. We all work together, postdoctoral researchers, and so they have the expertise from across the spectrum, from faculty members to full-time researchers, and they work alongside it, and it's really important. I don't like creating artificial projects just to keep people busy. It's 2 ways. I want to invest time in student work that is going to be useful for our research program, and that alignment is absolutely the case. And one of the reasons why Southampton is a serious university, we take it seriously. We do meaningful stuff with undergraduate students.
Yeah, absolutely. There were a couple of questions there that asked. There was actually one question from Emma, which was one thing. What would be one thing you would say is one of the best thing about studying biochemistry, particularly in Southampton? And I said I'd pose the question to you, but my own opinion was the fact that you're working with scientists. You're the scientists of tomorrow, but you need to be working with the scientists of today who are not making a pretend project for you, but are simulating you. You're joining their lab. You're kind of experiencing what real research is all about, and then you're being published. I mean, Maddie Newby, who Max speaks about, she's just finished her undergraduate degree. She's not yet even started her PhD, and yet if she's on 3 papers, that tells you that the research that she was part of when she did her 4th year project, or it would have been a summer studentship, because you're able to work in the labs in both of those situations, she was obviously part of research that was really important. But Max, there's another 2 questions that perhaps relate more directly to your talk, which I'd like to.
Okay, I'm also happy to add to that.
Okay, please do add to that.
Because I think, like with all research-intensive universities, the staff are chosen for mixed reasons. Yes, they're educators, but they're also researchers, and they do need to be really good at research. And so what you get is a big mix of people with really different skills, but they're world class in something. And that's incredible, isn't it? I mean, so, you know, you're taught by a whole range of different personalities and all the rest of it, but they've all got something to offer. And so I think that's one thing that I'd really like to impress on everybody is, you know, if you go into it with an open mind, you can, you know, really take home some world-class insights and approaches. And yeah, it's in that sense incredibly exciting.
Absolutely. I can't stress that enough. As a scientist passionate about what I do, I can't stress it enough how it's really important for us to get you guys to be excited too. But I'm digressing because there are 3 related questions, Max, that I'd like you to give us your thoughts on. So the first one is from someone who's anonymous, and then there's Devon Dali and someone with the initials SM. They're essentially related to the vaccines. So the anonymous question is, please ask Professor Crispin about how the different types of vaccines work. For example, the COVID-19 vaccine, how does the Pfizer messenger RNA one work differently to the AstraZeneca one? And then related to that, the second question from Devon Dali is about the degradation of the artificial messenger RNA once it enters the body. And then the SM is asking, how are these viruses actually generated? Because they're not exactly alive. So I thought 3 questions related to you.
Yeah, there's quite a lot there. So, okay, well, stepping back a bit then. So the major classes would be like in China, they have inactivated virions. So they use paraformaldehyde to inactivate, you know, otherwise infectious virus. And that's quite historically, you know, good way of taking it. You just take a pathogen, inactivate it, and use that as a virus. The ones more common in the UK are things like the Pfizer, which is RNA based. So that just gets the genetic instructions already transcribed into RNA, and it's formulated with lipids that I believe are manufactured in the UK, and those lipids help deliver the RNA signals, the information encoding the viral spike into cells. Now with the adenoviral system, there you're using adenovirus to achieve basically the same thing. But in that sense, it's all the genetic information is packaged into adenovirus, which then does the delivery. And then the other formats are recombinant proteins, like I believe the Janssen formulation, where all that manufacture, you're not doing it in your body, you're doing it in the lab. Now, what's been quite incredible are the adenovirus systems and the RNA-based, you know, the Pfizer systems, were, you know, space age, you know, new vaccine technologies. The ideas had been around, but they hadn't been implemented in quite the same way. And it's been a really incredible thing to see just how effective they've been, because there were question marks until you do the clinical trial. You just don't know how effective they're going to be. And what I find quite amazing is that they're all they're fairly comparable. You know, so within the UK, the clinical trials aren't showing massive differences. So yeah, they're all achieving the same thing, which is delivering viral glycoproteins to your body to elicit an immune response. And what was quite amazing is just the different strategies that can achieve that. So hopefully that answers that one. What's the other aspects?
So there were questions about they're not viruses are not exactly considered to be alive. So how have scientists been able to create a virus? And related to that, an anonymous question was, how did they come to exist? Where did they originate from?
So yeah, that's a really good one. So when you're manufacturing these viruses, you're actually doing it in cells. So viruses can only really propagate and replicate in the context of cells. So how these, like the AstraZeneca vaccine is created, is the components are all manufactured with cells, but it's not the cells that are used, it's only the products, so the adenoviruses which the cells have made. So that's how the virus is sort of manufactured. And where they come from, well, this is a really interesting one because it's a chimp, you know, it's a ChAdOx, the full name is ChAdOx. The ch stands for chimp. So what they've done is they've taken a chimpanzee adenovirus. And it's an interesting question about why would they pick chimpanzee adenovirus, why not a human one? Well, I think there's one consideration is what immune response are there already to adenoviruses. So adenovirus is a common cold. So if we all used a human adenovirus and re-engineered that for vaccine, some people would have existing immunity to it, and it may, get in the way of how effective it would be if you already got antibodies against it. So by using a chimp one, you've got very little existing immunity, and therefore you can your body doesn't stop it from delivering its messages. So they're naturally occurring, and they've just been re-engineered for the vaccine purposes.
Okay, another question about the vaccinations is a question from AK saying, I'm wondering why is it that you had chosen messenger RNA as a potential vaccine for COVID-19 rather than other strategies, and if people wanted to read more about messenger RNA vaccines, what would you recommend? Anything you'd recommend that they read? But why messenger RNA as a vaccination strategy?
Well, I mean, I suppose I would probably take a different view on that, in the sense that what we've seen globally is the opposite, whereby loads of different vaccines are being pursued, not just RNA, and it's very much a sort of UK, North American perspective to consider the RNA as the dominant form. As I mentioned, in China, it's not. So there's a big range of different vaccine approaches. With where we have chosen it, so with Pfizer, etc., it does offer certain advantages in speed of manufacture. So when you're making proteins, you have to be really careful about what you've made, how it's purified. The quality control is arduous. With RNA, it's much simpler. And so one of the attractions of that whole format was actually the rapid, rapid production. So, for example, I hear that for mutations, they can pivot their production within about 6 weeks to match a new mutation. Whereas if you're manufacturing proteins, etc., it'll be, I don't know the precise time, but it'll be a lot longer. So it's very rapid. And as we've seen, it is effective. So, there are lots of advantages. The disadvantages are things like it's not very stable at high temperatures, so you need a -80 refrigeration or freezing network. So again, when you think of a global pandemic, it's not suitable for every part of the globe. But within the UK, we're well set up to do that. But in other areas, other vaccine formats will have advantages. And actually, the adenoviral vaccine doesn't require the low temperatures. So for example, that one has advantages for that. So yeah, they've all got different features and they have different advantages depending on the, lots of real world considerations really.
And Juwan has asked a question there. Why do certain vaccines offer a lifetime protection whilst others require booster shots for some time?
Yeah, I guess there'd be two sides of that. One is your immune response. So what degree is that maintained and needing to be sort of boosted, if you like? And the other one is the drift on the actual pathogen. Is it staying the same? And if the pathogen's changing, then you need to have different boosts with different immune responses. I think it's too early to say at the moment about the current coronavirus, what is the, because they're so new, aren't the vaccines? We don't know to what degree immunity will hold out over the long term. But it's likely that we're going to be seeing this so-called antigenic drift with mutations becoming more dominant and therefore having boosters be quite important to catch up.
Max, one of sort of built into a previous question that I read out to you was how this artificial messenger RNA is degraded by our body after it enters, because surely it's going to be there for a finite period of time. And once the antibodies are made, we've got sort of the instructions on how to make those antibodies. Yep. But that messenger RNA, I guess, would just be degraded with time. It's
a really good question. And actually, the reason why I particularly like it is it shows an element of insight. Often people ask how things are initiated. Very few people ask, how is a response turned off? What is the natural stopping mechanisms of all these things. And within RNA, I think it'll plug into the natural RNA metabolism. So you can ask your question, well, how is a gene turned off? What happens to natural RNA? How's it degraded? And I think basically it's going to be plugging into the natural degradation pathway of RNA that you have in your cells anyway. But yeah, it's a very good question. What is the length of time of production? I'm sure the answers to those questions are known. I just don't know it's off the top of my head.
Okay. So we've got other questions. One question from Anne, I particularly like this question, is what sparked your interest in glycobiology, Max? Did you know when you were their age, 16 years old, you wanted to do glycobiology?
Not glycobiology. I knew biochemistry, actually. So I was unusual in realizing that I liked, I guess, a bit of a cliche, isn't it? But I liked chemistry. And I was very interested in biology, and so it was obvious, the nexus between the two seems to be fruitful. But what I realised is I had quite patience for detail. So where some people would look at all the different amino acids and the different carbohydrates and just think it a chore to learn about them, I really quite enjoyed it. And when it came to the carbohydrates, it was at university where lots of my colleagues, I have to say, didn't always see past the detail and just how cool it was. Whereas I had the patience, and once you'd learn some of the enzymology and some of the structures, you could then realise that it gave you something to say about such a wide range of different biology. So when you consider every single protein on your cell surface, bar just a handful of exceptions, are glycoproteins, if you want to understand how cells interact, then carbohydrates give you a whole new layer of understanding. And it's quite a productive way of thinking. I was quite struck when I found out that blood groups were carbohydrates. I hadn't a clue. I'd learned in school about the ABO system and all, in tissue incompatibility, but these are actually different carbohydrates on the surface of your cells, and the immune response to those carbohydrates defines the blood groups. So it was examples like that where I realised it was really useful and was beginning to explain a lot of things which I'd previously, been aware of. But yeah, you can see it. I found it very interesting, and I haven't looked back since, really.
Great. You've obviously chosen to come down the path of research. And I have had a few questions about career pathways for biochemists beyond research. And there are questions there which I guess can also come from the biomedical, people who want to do biomedical sciences and biochemistry, I guess could be put into the same category. So people you did your undergraduate with, Max, can you tell us different routes that they've taken that have taken advantage of the knowledge and skills they picked up? Because you also said you're utilizing the writing skills much more than you anticipated. We need to,
So again, I can answer that on two levels. One is my contemporaries, where are they now? What type of thing they're doing? But also I've already seen students like that I've taught where they end up. So there's a few professions that you don't really know exist when you're at school and even at university. And one of which is intellectual property. That's it might seem quite a niche area, but people own inventions, and there's areas of law where it sets out, how you can claim ownership of inventions, and these are so important in the pharmaceutical industry. So if you invent a drug, you need to be able to state what you've invented and protect it. So many of my contemporaries have gone into intellectual property. Others have gone into different areas of the pharmaceutical industry. And again, even on a regulatory level, what are the sort of laws surrounding the regulation of drugs? And, it doesn't strike you as something you'd set out to do, but it's incredibly valuable, and they're having a huge impact. We also have a collection of teachers, university lecturers, bankers, investment bankers, accountants, even actuarial work, so like pensions, the full, a few army officers, the full range of things really. It's I think what strikes me is that biomolecular sciences is use that phrase, a broad church. There's home for people who are very biological, very highly analytical, mathematical, good with computers. And so what you see with your graduates is that they're, going off into the big wide world in all sorts of different areas. But because it's analytical and, very scientific, and it equips you to deal with bodies of information, it's really valued by employers. So yeah, some of my immediate students have gone into intellectual property. One of them runs an education company. Yeah, I'm just trying to think. All a wide range of different things, really. So I'm not sure how that answers the question. Really, it's everything you could ever imagine, really.
Yeah. So Max, another question, which I suppose you are better placed to answer than I am. So you did your first degree in Oxford, and then you've experienced being an undergraduate in Oxford, and then of course you were teaching there as well before you came to Southampton. So a couple of students have asked what the difference in biochemistry is between universities, and one particular person has asked, what are your experiences of how you were taught yourself in Oxford compared to how you teach in Southampton?
So there are differences, but I think the most important things are the similarities. So I was quite impressed coming to Southampton that the material you're covering is of very high standard, and actually it's taught, so you quickly found out, by a load of people, what percentage of our lecturers have come from these places. It's a high number actually are Oxford and Cambridge and Ivy League university graduates. So actually there's not such a big difference, really. So yeah, I think that's the main message, is biochemistries and biomolecular sciences are fairly universal, and the academics in these institutions move around. They're the same, they're drawn from the same group of people, and so actually what you'll find is the similarities are more striking than any differences.
Yeah, I mean, I want to add to that very quickly, because then there's 3 questions that are science related that Max should answer. I did my first degree in biomedical sciences at King's College in London, and then I did my PhD in Oxford, so after which I came to Southampton. So I've been able to see teaching in 3 different institutes, and the one thing that I really liked about Oxford, and I find reproduced in Southampton, which I didn't get as an undergraduate, is the support. So every member of staff is assigned about 4 to 6 personal tutees in year 1, and they then mentor those individuals for the 3 or 4 years that the students stay with us. And we do care that you succeed in your ambitions, and that level of support, helping you to make the right choices as you go along. If you want to go and do medicine, advising you about what it is that you need to get done. If you want to go and do work in industry, advising you about how you can apply for applications there, helping you with year in employment. I think that support system is quite important. Some people don't need it, but some people benefit from it. So that's one thing that I think is potentially different that you may want to consider. But Max, there's a few questions there related to the science. So there's a question there about how, so Professor Crispin, do you think a viral vector vaccine might be able to deliver more than one genetic material of a different virus to offer a wide range of immunities to certain diseases?
The answer is yes. So multi-subunit vaccines are of considerable interest and are attracting a lot of attention. Those aren't the ones that have been approved, but when you look at the what's been deposited on preprint servers, the non-peer-reviewed stuff, there's an awful lot of activity investigating precisely that. So whether you can incorporate them all in the same adenovirus, for example, well, yes or no, you could always just mix a whole load of different adenoviruses together with different genes in. There's various solutions to achieving that suggestion. So it's really good suggestion. People are actively pursuing it. And I'm, with the case of coronavirus, is fairly confident actually that some sort of multi-strain based vaccine would prove pretty useful. So yeah.
And I guess related to that is, there's another question. Is there one type of vaccine which allows you to build up immunity more quickly than others once it's in the body?
That's a good question. I mean, the proof there is down on the clinical trials, and the emerging picture there is, yeah, despite the variations, there's not such a huge difference. So yeah, at the moment, I would probably argue that the differences are not massive. I mean, once the protein's there, I mean, with the nucleic acid, you add in a few extra hours of manufacturing in your cells versus delivery. But on the grand scheme of immune response over a 2-week period, you know, there's no real big differences, I don't think.
And so one more question is, would these viruses, such as influenza's manipulation of cellular proteostasis machinery like chaperones, therefore influence the interactions of the viral glycans and the enzymes?
So is this in the case of a natural, presumably they mean in the case of a natural infection rather than as a vaccine?
Yes, perhaps.
So in the case of, and all the things I've talked about have been about vaccines, but of course within the normal biology of these enveloped viruses, the carbohydrates have a huge impact. So, and similarly, what the virus does to the cells can also have an impact as well. So we find that the carbohydrates that are presented can influence how the immune system is recognizing the glycoproteins. It can shape the immune response, and indeed the virus can perturb glycan processing itself. And now it's an area of active research about how significant that is. But yeah, these are highly complicated areas of biology, and so that's a really sensible suggestion. Yeah, multiple factors can influence the sugars.
The question earlier on was, what makes some viruses more contagious than others?
Yeah, that's a really good question. I'm not sure I could fully. This is slightly outside my lane of expertise, whereas we have people in Southampton who specialise in this exact topic. So hats off to them, but I'll do my best. So things like the stability of viruses outside of the body. So, you know, when viruses are deposited on surfaces, how long do they last? And that can be influenced by their composition. So, for example, if you're relying on a lipid envelope, you may imagine it being less stable than a dense protein coat. You know, you can easily see that chemically they're very different, and they have consequently quite different half-lives. Then you look at things like aerosols. You know, to what degree can they be incorporated in aerosols? And with that, you've got two sides of that coin. You know, to what degree can the viruses actually be be put in an aerosol on a sort of physical chemical basis, but also what cells are being infected, to what degree are your viral load contributing to secretions, and all of these things, you know, play in. So it's a multi-variable equation that then dictates about you know, contagion. And then there's of course the mode of infection, you know, so whether it can be done by touch or whether it's other exposure routes that are more likely, you know, that's all very different. So yeah, you could spend a lifetime pinning all these down.
So a question again from Devon Darley. Regarding the vaccine again, how do you feel about the possibility of allergies to the PEG-glycated lipid nanoparticles that are being used in the messenger RNA vaccines?
Yeah, well, from what I gather, I mean, just looking broadly, the complications from vaccines are currently very low. Even when we see, you know, some health events have occurred, they're still at a frequency that is, you know, really remarkably low. So whenever I think about these topics, I always think, well, it's not the absolute risk of, you know, adverse events. It's, you've got to weigh it up against what it's like to have coronavirus and the consequent, you know, risks associated with that. And for me, I'm currently unvaccinated. I'll be accepting any vaccine offered because I know that the sort of rare events associated with any of them are so much more tolerable than the actual consequence of infection. So I dodged the question a bit there, but in generals, that's how I'd approach it.
And we have a question from Ben. I think you've alluded to this a little bit, but let me read out the question. So what is the advantage of using messenger RNA in the Pfizer vaccine if it is less chemically stable than viral DNA, being only a single-stranded molecule and having exposed bases, meaning that the Pfizer vaccine has to be stored at lower temperatures? What's the advantage in having something that is less chemically stable?
Well, it's really good at inducing gene expression, and so you're getting really good production of your glycoprotein. And so they've not settled on this at random, so it just happens to be very effective. So if you do have the cold storage distribution network, then it offers quite a lot of advantages. Okay,
I'm just looking back at any questions that I didn't ask you. Okay, I think I've asked you most of the questions that related to the vaccination program and your talk. Yeah, I think I have answered most of those questions. So there's a question there from Anna, which is about the difference between integrated masters and doing a version of it that's not the integrated, the bachelor's version, about whether the last year is more focused on the research project, and if you're interested in that, can you choose research in the cancer field? We have many scientists within biological sciences that work on cancer research, just as Max works on glycobiology and I work on Alzheimer's disease. There are many other individuals in biological sciences who work in different areas, including cancer research. We've got colleagues working on breast cancer, other colleagues working on contagious cancers. Even if you're coming to, we have a visit day, virtual visit day tomorrow, and one of our program leads, Professor Hannah Siddle, she works on contagious cancers in animals. So there is no reason why, if you did a master's project and you chose one of those members of staff to work with, why you couldn't go and work with them and do research in that area. And so the integrated master's version of any of our degree programs essentially offers you everything that's the same as the undergraduate version of it, the bachelor's version of it, up to year 3. And then in year 4, you stay on and spend half the year working in a research lab. And you do pretty much get to grips with what it's like to do research, so that if you decided to go on to do a PhD, you have a very good idea. And like Max, a lot of times I select students for PhDs because they've spent a year in my lab and they've already learned the skills and they've already shown me that they're passionate and they've learned everything, and so they can now apply their skills to answering the questions. So that's really one difference. I think we've answered most of the questions there. If you do have any more questions, I'm going to put my email address there. Yeah, so do email me, even if you have further questions. Okay, there is a question there, Max, that I didn't see. It's a question that was posted by an anonymous person. How does developing vaccines for newly emerged viruses such as SARS-CoV-2 differ from those that are well established but have lots of variations like HIV?
Yeah, so good question. I think the main one is just how much more, well, it's more simple really, isn't it? Because there's just one or a limited range of viral isolates. And so one of the messages from my talk was really how all the techniques being developed for HIV were just moved over to the coronavirus in this case. So it wasn't very different. It was, in fact, almost identical. The biggest difference was that it worked with coronaviruses, whereas HIV, because of the variation, those same techniques are not so fruitful. But it's the same people, it's using the same techniques. It's just one's productive and one gets a solution and the other one is still eluding us.
I think the silver lining of this pandemic, if we can call it that, is it really demonstrated to society how knowledge and skills in disparate areas can come together to solve a problem that is a real challenge to society. So exactly as you said, Max, what was known for one type of virus, one type of vaccine, respiratory research, it could be disparate areas, but actually they can come together. And this is how the future is. The future is interdisciplinary. The future is not being married to a discipline, not being married to a particular way of solving a problem, but perhaps being more open to using any different method that works in solving the problem, being open to utilizing different interdisciplinary methods to solve the problem. I think I'm going to take one last question because otherwise it'll be unfair. It was put up there at the same time as the last question you answered, Max, which is why do some vaccines have longer term consequences and others don't?
Yeah, by consequences, I'd probably need to sort of pin that down. So longer term consequences, that has connotations of being negative, but it could also be positive. Yeah, in terms of the impacts on the immune system, multiple things are going to influence the half-life of the immune system. In terms of negative consequences, like adverse events, those are going to be particular to that particular vaccine format. And what we're seeing, like with the rare clotting conditions, they're beginning to pin it down to particular features of that particular vaccine strategy. And that relates to particular B cells that make antibodies and which ones are triggered and which ones aren't. So sometimes rare types of antibodies could be triggered, leading to rare events. So it boils down to the details. It's always unique to a particular vaccine format what the tail risk might be. But again, I want to finish by emphasizing just how low those rare events have been, and it's really a very uplifting story in the case of vaccine development.
Thank you very much, Max. Both Max and I were slightly nervous that we would be sitting here without questions to answer, and we'd actually made a couple of dummy questions for me to ask him in case you guys weren't interested. But I'm so pleased, so very, very pleased that you've engaged with us in the way that you have. You've asked us some questions that clearly show us that you care about this area, you're passionate, you think. That's what we want. We are passionate about what we do, all of us are, and we want to pass that passion on, that knowledge. It's not just knowledge, you can get the knowledge from textbooks. In Southampton, what you get is something beyond that. You get some of the passion. One flame lights other flames. You get part of that passion, and you get to be part of the movement that decides to make a change. So I'm really happy that you joined us today. I hope you enjoyed the session. I hope you learned something. I hope you leave the session knowing more than you joined. And if you do decide to come to Southampton, or if you have any further questions, do drop me a line, even if these questions relate to something that is to do with a vaccine, because I can always pass them on to Professor Crispin, and I'm sure he'd be happy to answer them. And do look out in the Daily Mail for further insights from Max's work. But thank you very much, and have a good evening, everybody. Goodbye.